start discussing this a little bit. Today is the 25th? Okay, 2018. So, you built some DNA. Do you feel at this point like you have a little bit of a sense of what the DNA actually is?
Yes.
A long molecule composed of 5 chemical pieces. Every living thing uses it to pass on genetic information. The next big question we've got, because we kept saying that mistakes in copying led to differences in DNA, and that those differences in DNA, in some cases, ended up protecting bacteria. So what we want to know is, how does it actually get copied? The process of copying DNA is called what? Do you know? It's one of your vocabulary words. No, DNA copying is called replication. Okay, a lot of you got it. It's the second word on the vocabulary sheet. We can talk about replicating anything. We can talk about, I'm going to take this paper to the copy machine and I'm going to replicate it. We don't usually, we just say copy. But replication specifically is the process of copying DNA. When does DNA replication have to happen? Talk to your table. When does DNA replication have to happen? It's happening right now. Okay. So when you reproduce, you're saying? Okay.
Wait, what was the question?
When does DNA replication have to happen? Okay, so I got some interesting answers. A few people said, well, when you reproduce, when you make a miniature version of you. Yes, that's true. That's a special kind of replication that we'll talk about later. Every time you make a new cell, the new cell needs all the same DNA the old cell had. Now, if you happen to be a bacteria and you're only one cell, then making a new cell and reproducing are the same thing. Because that's all reproducing is for a bacteria. For multicellular organisms like us and this plant and an earthworm and a dragonfly and a fish, it's a little more complicated. Go ahead.
When does it replicate?
Every time you have to make a new cell. Replication.
I was right.
Happens, yeah. Every time you make a new cell. Every time you make a new cell, you have to copy the DNA so that the new cell has all the same DNA. Now, let's draw our DNA. Okay, so we've got our DNA, and we want to copy it. Now, there's a convenient fact, which is that each half is basically a template for the other half. But the first thing we have to do if we're going to copy this DNA, we can't copy it all twisted together and joined across the two backbones. It has to be unzipped. So step one is literally unzipping the DNA. Okay, now there's a lot more technical detail to this than we're going to go into. When you're going to do some little videos on the process of DNA replication, and I've got one that's got a lot more technical detail and one that's kind of basic. We're going to go through the basics here together, and then we'll talk a little bit about those videos and details. So the DNA actually gets unzipped. Anybody notice a difference between my complete DNA and my unzipped DNA? Did I go all the way down? Well, there's no twist in it, obviously. It has to sort of uncoil in order to get, and we could take this back down like that. But I only am replicating it to, what, here? I'm only doing a section at a time. And DNA gets replicated a section at a time. Because you can't, think about this. We said there are miles of this stuff in your cells. You couldn't replicate the whole thing all at once. Pardon? Well, essentially, there are a lot of your cells in the moment before one dies. So, you are reproducing cells. But no, we can't deal with this whole thing at once. We have to copy a section at a time. On one side, and there are some differences in how it's done. On one side, those sections get joined. They're little fragment pieces. On the other side, they do actually work in a continuous strip, but it still has to go a section at a time. Okay, so we've got it unzipped, and now comes the really interesting part. Okay. Does anybody remember what this area is called from the pictures you drew? It's a fork.
The
Replication fork. Oh, because what's the process that's happening here? Replication. Oh! If you see a fork in the road, take it. Don't take the spoon. Ah! It's not funny, but I do my best. So that area where they're splitting is called the replication fork. And you don't have to know that. What's going to happen next is a new strand of DNA, a new half of a strand is going to be created here. Guess what's going to happen on the other side? The same thing! We're going to get 2 strands of DNA out of 1 old strand, which is pretty cool. And this is happening in your body right now. Yes. So, what I'm going to have you do at this point is you're going to go, once you finish drawing this, you're going to go back to your partners, and I'm going to hand you a new sugar phosphate backbone. And you're going to go through the steps of DNA replication. So let's just quickly kind of review this. So we unzip it. We insert matching bases. And what we end up with then are actually 2 new strands of DNA that are identical to the first strands. Whoops, shoot. 2 new strands of DNA that are identical to the new strands. So 2 new DNA strands. And then those can go into new cells. So this process happens every time a cell reproduces, which is happening all the time because you're getting new cells as your old cells sort of age out. So you have 2 new DNA strands. Okay, finish copying this and then go back. Oh, and then of course, I forgot to mention, these things join up and then those get retwisted. And you drew the picture of this. You drew the picture of replication and you saw that there were 2 new strands. So when you finish drawing this, I want you to go back to your partners. I'm going to give you a new sugar phosphate backbone. If you're not totally done replicating your DNA, that's okay. That is some beautiful DNA, sir. What I want you to be aware of, if you're not done, just hold for a second. This is so what does it mean to conserve something? To save it, to not use it all up. DNA replication is called a semi-conservative process. What does semi mean?
Half.
Like halfway, yeah. So if you look at this new DNA, half of it is recycled from the old. Half of it is new. So it is a can't fit that in that space, semi-conservative process. Now, if we replicate this DNA right here, the next time, if we imagine that green is now our new DNA, what are we going to have? We're going to have 1 strand that's green and a dark side, and 1 strand that's got 2 new sides. Oops. because every single time it gets replicated, half of it gets saved. Half of it's brand new, but half of it gets saved because it's a semi-conservative process. So eventually we're lacking this whole old section there. Okay, you can finish building your DNA. When you've done that, I want the cups up here. And you get to keep your DNA. I would like you to keep it for the rest of the year. Tuck it into a notebook safely. Tomorrow when you walk in, there will be a recall quiz on DNA replication.
Wait, what's that say?
Semi-conservative process.
I can't.
If I can just take one moment to revisit. I take a moment to revisit that. Do you feel like you have a sense of how the copying goes, how the replication works?
Yeah.
So tomorrow, I'm going to tell you this. My personal approach to life, and some people think this is a little neurotic, but I don't. It works for me. I like to actually think about the worst possible thing that could happen and then figure out how I deal with that. Just because I know the worst possible thing probably won't happen, but if it does, I would really like to have thought it out beforehand. So I tend to think like, well, what would I do if my car went off this bridge? How would I get my child out of the car? How would I get out of the car?
I've thought of that.
Yes, it's good to think about those things. It's good to think about those things. So tomorrow, we are going to think about what can go wrong with DNA replication. What can happen in that process that can cause a problem? And you're going to have a little video work to do.